Okay, thanks for the introduction. Thanks for the introduction and thanks for having me speak here. So today we'll be talking about KeyGene's journey into Apache Spark and how we use it to run our genomics analysis. So an overview of this talk, to start out, I'm not really a data scientist or a computer scientist, so I'm a scientist in computational biology, bioinformatics. So this is not really my comfort zone, but I will try and manage my best. The scope of this presentation, so I will be starting out, let's say, KeyGene's journey into Spark, so how we can use this as a tool to enable high throughput, high-speed genomics. So it's told from a user's perspective rather than from a developer point of view. And our goal is to enable a kind of interactive analysis of genomic data, so genomic data processing and querying. So the contents of this presentation, I will give a little bit of an overview of KeyGene as a company, what we do and what our aim is. Then I will give a crash course genomics, because I can imagine not many people here have a very deep knowledge about genomics. And then I will talk about the big data challenges that we see in genomics and how we solve them with Spark. So to start out, when we look at the world, what we see a kind of global trends. There's more and more people on the planet with less and less resources. So there's climate change that drastically changes the planet that we live on. There's less land, less water, less fuel available to feed all these people. We see that there's more illnesses. There's obesity. There's also malnourished people. So what we end up with is that we really need to focus on yield. So we need to improve the yield of our crops because we get more and more people, the same amount of space. So we need to improve the amount of food that we can grow on this amount of space. So going from the 1960s up to the 2030, we will double the amount of people on the planet. So that means on average we have half the amount of land available to grow the food that we want. So as KeyGene, our most important focus is the genetic improvement of crops. So to make sure that we can achieve this higher yield, but also to achieve better vegetables, better fruits, and we achieve this through molecular breeding and also molecular mutagenesis. So it is important to know that this is not a GM approach. So the approach that we take is to use the natural available variation and also to induce novel variation and then use this in breeding programs. So a little bit about KeyGene. We are around for about 25 years now. And we started out as a technology company that was founded by several different breeding companies that wanted to join forces in order to develop new technologies to help them out in their plant breeding. So within these 25 years, we have grown to the go-to AG biotech company for higher yield and quality. Our current shareholders are 4 large seed companies, 2 Dutch companies, Enza and Rijkswaan, and a French company, Vilmorin, which is part of the Limagrain vegetable seeds group, and Taki Japan. And so we work for these 4 shareholders, but we also work on what we call the field crops, so the non-vegetable crops for the rest of the world. So our research and development strategy at KeyGene focuses, let's say, on the field where you would say universities on the left-hand side and the breeding companies on the right-hand side would kind of meet. So we try to develop novel technologies and novel traits. So not so much focus on the fundamental research, but more on the applied research, let's say the pre-competitive research for our shareholders, but also the specific projects that we perform for non-shareholder companies. And yeah, we like to position ourselves so in the middle and have interaction both also on the academic side, but also on the companies that we work with. So to place a little bit my story into perspective of big data, I work in genomics. And what we have seen over the last couple of years in genomics is a tremendous growth in the amount of data that's being produced. So as you probably all know, the human genome was sequenced in around 2000, 2001, and this was a very large project, a multi-billion-dollar project. It was very expensive to generate this genome sequence, but over the past 2 decades, we have seen a tremendous decrease in price for this sequencing. So this technology has developed a lot with the scope of, with the aim of getting to this $1,000 genome, this promise where you can sequence a human genome for only $1,000, and we are close to achieving that within the community. So you see that the amount of data that we can produce also is growing, let's say, at an exponential scale. So this is a log scale, and you see a kind of linear increase in the amount of data that's being deposited into these public repositories. So these genomic sequence information that is present. So our challenge is now, how are we going to deal with this rapidly growing amount of data, whereas our compute power and our resources to analyze these data don't scale in the same way. Well, like I said, KeyGene is a plant breeding company, and I come from a plant genomics background. So to highlight a little bit the differences between plant genomics and human genomics, when you deal with human, you have one genome, so we all share more or less the same genome with some variations between them. And because, let's say, 90% of the genomics field is working on working on humans, you also see that there is a very nice shared resource there. It has very simple genetics in the sense that, I mean, you have two parents, so you get one half of your chromosomes from your one parent and one half from the other parent. Whereas with plant breeding, you have many crops that we work on, so each crop has their own genome, and they have a lot of more complex genetics. So there's, I will go into a little bit more detail, but there's a lot of backcrossing, there's a lot of polyploidy, so multiple copies of the genome present. So it is not as simple as human. Another problem, again, is that within the plant world, because we have so many species, but because there's also so many different interests, there's an enormous fracturing of all the resources that we have available. So we cannot bundle all our forces and work on this one genome as has been done in human over the past 20 years. But everyone is working on their own favorite organism, their own favorite genome, using their own tools and data that they have available to them. So yeah, the quality of our resources is a little bit lower, it's more variable, and it's also more fragmented what we can access for each of these individual crops. And then finally, the way that we apply genomics in the plant field is different, of course, than from the human field. I mean, in the human field, it's more as a diagnostic tool. We want to measure and understand diseases. We want to improve the health of people by looking at their genome, having personalized medicine, those kind of things. Whereas for plants, we actually want to direct how we breed and grow those plants. So we want to create new varieties and improve what we have. So we're using genomics as a tool to improve, let's say, the yield of our crops, or to improve the fruits of our crop. So we can direct our efforts using this. Now, to give a little crash course on genomics, let's start out with plants, because that's what I work on. We deal with a wide variety of plants, and they have a wide variety of genome sizes. So to put this a little bit in perspective, human is about there. So you have a 3 gigabase genome, whereas with plants you can have from a few hundred megabases, so this is the amount of DNA in the plant, up to tens or even sometimes hundreds of gigabases in the genome. So in that sense, human is actually peanuts. It's about the same size as the peanut genome, actually, but peanut is a little bit more complex than human. So, and now if we look at the different properties of the genomes, I mean, the human genome is diploid. You have one set of chromosomes from your father, one set from your mother. But from plants, you have had, over the course of evolution, many genome duplications. So you can have, for example, tetraploids, where you have 4 sets of each chromosomes, or even hexaploids, where you have 6 sets of each chromosomes. And this also makes the application of genomics, so assigning data to a certain copy of this chromosome, much more difficult. Now, I've been talking about chromosomes and DNA a lot, so if we look at this from let's say, a data science point of view, DNA, we can represent this simply as a very long string of only 4 different characters. So we have an alphabet of 4 characters. For some reason, biologists chose A, C, G, and T, because these are the abbreviations of the molecules that they represent. And so DNA is then grouped into chromosomes, and each chromosome is this very long string of only these 4 letters. So these chromosomes are millions or sometimes even billions of nucleotides of these letters in length. Now, we have technology to read DNA, but we only can read short pieces of DNA. So at the moment, we are limited to pieces of 100 to a few thousand nucleotides that we can read at a time. So it is not possible for us to read the entire genome of an individual in one go, but we can read little bits and pieces. And this is one of the challenges that we face on how to deal with this data. So if we now look at a genome, and I zoom in a little bit, you see a little piece of sequence there. So this large bar at the top represents one of the chromosomes in the genome, and this is just a small fraction of it. Now, well, this genome is not only these letters, because these letters actually have meaning. So the meaning in the genome is what we call the genes. And the genes are just intervals on the genome that have a specific meaning or function related to them. So for example, you could have a gene here that is relevant for drought tolerance, and you could have a gene here that is relevant for fruit size. And we basically represent these genes as a kind of intervals on the genome in a very simplistic view. Now, what we are interested in, of course, is to find differences between individual genomes. So, for example, if we have a genome here with the two genes you just saw, and we have another individual with a very similar but slightly different genome, we're interested in these little differences between those two, because these differences can have a kind of impact. For example, if we are looking at the gene for fruit size, this individual could have very large fruits, and this individual could have very small fruits. And that is determined by these variations in the genome, the polymorphisms as we call them. So and there could also be polymorphisms anywhere outside the genes, and they generally have no impact on what the individual will look like. So what we generally want to look for is to use these data to find the differences between the individuals. Now, one of the ways we do this is we generate these very small pieces of sequence, and we then, in a process that we call alignment, try to identify the location of these small pieces on the genome. So we know what the genome should look like. We have our very small pieces, and we try to kind of in a string similarity search kind of way try to find where these pieces should end up. And if we then we have to deal with measurement errors because the pieces that we can read are not perfect. We have to deal with true variations that are actually the differences between those individuals that we are looking for. And yeah, there's a lot of complexities in the genome, like repeats, that make it very difficult to accurately place each of these little pieces on the genome. But once we do this, and we can make a kind of pileup of all these different reads, different regions of the genome, we can indeed identify these differences that we're interested in. And then these differences, again, cause the difference between large fruits and small fruits, for example. So now we have our genome, and we produce these reads. And this is where it comes into the domain of big data, because we don't just produce some reads for a genome, we actually produce a large redundancy in reads for this genome. So about 30-fold or 50-fold redundancy in order to have a highly accurate representation of these variations. Now, we don't do this for a single individual, but of course we do this for many individuals. So if we would start out with, for example, a 5 gigabase genome, and we can read little pieces of 100 or 200 nucleotides, then we end up with billions and billions of these pieces of sequence that we want to compare to our genome to find all these differences. So, and then what we expect is that it will be potentially up to hundreds of millions of differences between individuals. So, to recapitulate what I just told in a few simple lines, we have our genome as a kind of text representation of only 4 characters. The variation is what we're looking for, so we're interested in how these different individuals differ from each other and how we can associate these differences to the traits that we're interested in. And yeah, we can read the genome in little pieces. So we have high throughput, highly parallel machines that can generate these data quite rapidly, actually. So it takes about a few days to read a genome in all these small pieces. And then we can do this for hundreds or even thousands of individuals at a reasonably low cost. So given for a few $1,000s per individual. Now, genomics data analysis is challenging. So we see that there's a rapid increase in this data generation. So 5 years ago, I wouldn't be talking about sequencing 1,000 genomes. I would be talking about sequencing 1 genome, and it would be a challenge. And now we can routinely really sequence hundreds or thousands of genomes. So this also means that there's a rapid turnover in the technology, because this is scaling up, and every time they come with a new device, and this gives new types of data, new ways to represent the data that we want to work with. So it makes it challenging to analyze these data. And yeah, this is a very scientific field. These are people like myself coming from a molecular biology background who then write the tools to analyze these data. Well, we're not really computer programmers, and this is not only my personal opinion, but also the opinion of the community in general. This scientific software is just bad. It's just full of bugs, doesn't work, isn't reproducible. So, but this is the world that we have to deal with. So the way we generally tackle this is by using high-performance computing. So the challenges that we have in front of us, we need to align these billions of reads to the genome. Then we need to call all these variants from these pileup of reads relative to the genome that we know. And then we want to identify these 1 or 2 variants that make the difference between a small fruit and a large fruit. So we have to go from billions of data points to millions to, let's say, 1 or 2. And the way we have done this over the last 10 years or so is by using kind of classical HPC infrastructure such as SGE clusters to run all these jobs in batches, analyze all the data. So really in a kind of non-interactive way, in a way that you have to think about your entire data transformation from start to finish, program this out and run it over your cluster. And of course you have to manually deal with all the failures and all the problems that you run into, all the crashes. So what we see is that there's many software tools that we use are actually kind of monolithic pipelines that perform 5 or 10 steps in one go, and they don't give you any control over these tools. So you run this on your SGE cluster, and you start praying, hoping that it doesn't crash, and at the end you get your result or not, and you have to start again. Well, one of the things that we see when we use this kind of classical SGE type of infrastructure is we have to buy an expensive storage server, we have to buy an expensive cluster, we have to buy an expensive network to connect actually all these pieces, and it has to be highly reliable because stuff cannot break, because then you have to restart your analysis and figure out what went wrong. And on the other end, we are now looking at Spark, and we have a commodity hardware that we can scale almost indefinitely at linear costs, and that is fault tolerant, where jobs can crash, and Spark will fix it for us. So this is very interesting from the type of analysis that we want to do to use this type of hardware and software. So what we started out then at KeyGene was looking at, let's say, this application landscape in genomics. So typically you have a kind of division of a lot of jobs that you run on a traditional compute cluster, so that are highly parallel, that require a large data processing. And you have jobs that require a whole bunch of memory, where you need to take all the data, put it into memory, and then do a kind of cross-tabulation of all the data against each other, for example, and end up with your result. Then over of course, what you see is that there's also a growth towards, let's say, dedicated hardware solutions, GPUs or FPGAs, to solve a specific piece of the pie, let's say, a specific subset of problems that we deal with. And we see slowly now that there's also applications coming from Spark. And actually what we're looking for, so can we leverage this kind of cheap hardware, this kind of scalable hardware, to also go into this into our domain, so into the genomics domain. So over the last year at KeyGene we have been looking at this. So the challenge that we see, of course, is we have these this rapid changing field with new technologies, new tools coming in all the time. The tools are built around files, so we have to deal with files rather than streams of data or data sets that you can store in memory. You have to read from a file, so you always go from file to file to file to your end result, which is also a file. And yeah, for example, the general type of workflow that we have is we start from our sequencing apparatus, which produces a bunch of text files that contain our data. Then we transfer, we compare this to the genome that we have. So again, we get a text file and another text file, and then in the end we end up with our variations, which are again a text file. So we have a lot of file-to-file transformations. Now, we started trying to see if we could solve this in a kind of more big data structure. So using Spark to perform these analyses, and so we started building our pipeline. So using Scala to wrap some of the tools that we have, because we're still dealing with these legacy tools that actually expect files in, expect files out, but still perform all the steps as much as possible on our Spark cluster, so that we can leverage this infrastructure. And so starting from, so some of the problems that we run into, for example, our DNA sequencer gives a whole bunch of files. And actually, these files are structured in a way that you don't get one record per line, but you have multiple lines per record. So we figured out we have to extend the default input reader from Spark and make it so that you can read multiple lines and get your records. But another problem is, of course, the sequencer doesn't output one batch of files, it actually outputs two batch of files, which are interrelated to each other. So we read two pieces of DNA at the same time, and they're connected. So to solve this issue, again, we had to kind of interleave these data with each other. So build an RDD where we can store these strings, so these strings actually, which are 4 strings already, and then interleave them such that you get one object or one representation of a single pair of data points. Well, we have all these bunch of files, and we can now store them into a nice RDD. And then, of course, we can run our mapping, so our alignment tool on this. And this is basically a script that we had to. This is a tool that we had to wrap inside a little Perl script, because our tool can only read files and write files. So we can use pipe, but then the tool doesn't understand it. So we made a little Perl script that actually writes it to the local file system and then spits the file out back to the local file system and pipes it out again. So it's kludgy. It's not where we want to go, but it's where we have to go at the moment, because I mean our tools are what they are. And then from there we can again take all these data together. We have to put a header to it, because that's what the file format dictates. I mean, bioinformatics has peculiar file formats, so they want headers on their files and not single line records and those kind of things. And then we can use this file for the next step. So I had hoped actually this morning to have to see a very nice keynote talk about Hill. Unfortunately, Cotton couldn't make it. So, but yeah, there are many tools already to deal with, let's say, the more downstream application in genomics. So finding the variants, but also processing these variants and then trying to find the interesting ones from them. So this is what we call genome-wide association. So linking the phenotype that we observe, for example, the difference in fruits, fruit size, to the difference in genotype that we observe, so these variants in the genome, and then trying to correlate which variant actually causes this difference. Because if you just measure enough of these different individuals and you see the difference in fruit size and the difference in their genome, you will in the end come out to a single region that actually defines this difference. So yeah, like I said in the keynote talk that wasn't there this morning, you would have learned a lot about this. So I purposely skipped this part in my presentation. We also worked on because Hill wasn't there at the start of this year. So we also worked on our own kind of system to deal with these data. So to start from our text-based variant file and then convert this into a Parquet file, going to an RDD so that we could actually perform SQL type of queries on it, which we are quite familiar with, to analyze our data and to select our data, and in the end get to a subset of data that is interesting for us to analyze further. And of course, once you have these data, you want to combine them with the phenotype data, so these differences in fruit size, for example, and do this genome-wide association on it, so that you can see actually which variants cause the variants. Now we have looked now at a bunch of these tools, and we try to implement them in our Spark ecosystem. Of course, the vision that we have at KeyGene in big data is larger than that. So we have focused now mainly on this first little balloon. So let's say the straight up genomics data processing, going from data that comes from the sequencer to the variation between the genomes. But of course, you also want to look at things like expression or phenotyping, or even consumer data that you can all feed into this system. And then we're aiming to get this big black box that we call big data analytics. So a kind of Spark framework that can integrate all these data, and in the end give us the genes that we're interested in, that are candidates for the trait that we are observing. So, and with this in mind, I would like to wrap up the presentation. So what I've shown today is let's say our first journey into the first journey of KeyGene into big data, and to use these tools in order to really accelerate our processing, massively parallelize the things that we had to do on SGE, to do this now on a Spark infrastructure that we can scale indefinitely and linearly. I also showed you some of the challenges that we dealt with. So the domain of genomics is quite focused on, let's say, file in, file out. So there's not many applications yet that make efficient use of the infrastructure that we have available. And yeah, I'm looking for a community there. So I'm looking at all you guys. Maybe you guys know something about genomics or are interested to learn about genomics and have a talk with me to see how we can actually take these tools that we have, these old-fashioned file-dependent tools, and put them into this new and very interesting framework. So to accelerate genomics with us. And with that, I would like to end the presentation and take your questions.
Anyone have any questions? So I get to you. Could you hand me? Oh.
Okay, first of all, thanks for the talk. On a few slides, you mentioned the differences between the human genome analysis and the plant genome analysis. So there's a movement right now going on called Thousand Genomes Project, where they go away from a single human genome, but want to sequence different populations around the world. So I wanted to do the same for tomatoes, for example.
Yeah, so there has been recently a very large project, 3,000 rice genomes, where there was a public effort to sequence indeed 3,000 rice accessions. Of course, the difference there is that when you talk about the human genome, you're talking about a very tightly related group of individuals. Whereas when we talk about plant genomes, we also, within our breeding, we like to use, for example, wild varieties. So let's say it's more. It would be more similar to the 1,000 primate genome. These type of projects, because we're looking at a much wider genotypic diversity. But indeed, we're in plant community, we are doing these things for sure.
Any other questions? Yep. Oh, question? Now come back. You have a question? Yes.
Thank you very much for the talk. I have one question about how you use Adam in your pipeline. How do you store BAM files and how do you query them?
So at the moment, we actually do not, within the pipeline that we developed, we do not use Adam. We have played around with Adam to see how we can use this to indeed query and index all kinds of files. But yeah, at the moment, it's not included in the pipeline.
There is a blog post online of how Spark and Adam works.
Yeah.
If you Google it, you should hopefully find it. If not, come
Yeah,
find me.
so Adam has a very nice amount of APIs to query and access your data, but it doesn't have many tools yet built on top of this API. So you can store your data in a very nice format, which is very compatible with how Spark wants to access your data. But then, yeah, we are stuck with tools that want just POSIX files, that want flat text files to put them in. So in the end, you have to write them again to that. So we simplified it. At the moment, we have a more simplified workflow where we indeed just put our data into an RDD and then write it again to a text file.
Another question?
So I have another question. Okay, regarding your genome analysis, do you actually consider, it's related to the first question, why to sequence multiple genomes, so you can make a lineage analysis, so you can track the epigenetic changes from the wild type to the industry types. Do you have considered such approaches as well, where you also take into consideration how the genome changes due to influential weather factors at different locations, for example?
Yeah, for sure. I mean, the approach that I show here is more of the high throughput approach, where you sequence tens, hundreds, or thousands of individuals from a certain species and want to just find differences. Of course, there's more interesting differences, like indeed changes in genome structure. But there you also have to use different types of technologies in order to get those data, which are much more expensive at the moment. So we cannot really do this at the scale I presented here yet.
Have you tried approaching any of the developers of the most common bioinformatics tools to Sparkify their algorithms? And if yes, what was their reaction?
So I have not approached any developers directly. We have had a very interesting Spark session at the European Bioinformatics Conference this year, where there was indeed also a call to the community to develop these tools. So what you see is, like I said, bioinformatics is kind of a field where people mostly have come in from the biology side of things, and then teach themselves how to script, how to program, build these tools, and then run them on classical HPC infrastructure. So there is now an influx of people coming from more from the computer science field into bioinformatics, and who are really developing software. So it would be very interesting to get in touch with those people to see if they can indeed develop their tools on top of Spark.
Cool. Thanks everyone for attending. It was a great talk. Everyone, round of applause.